prophase, metaphase, anaphase, telophase. How do these stages relate to mitosis and meiosis? Hello, SciPals! I am Sir Jerry Lagmador, your science teacher for today. In this episode of Agham Alam Hub, we will compare mitosis and meiosis. We will also discuss the role of mitosis and meiosis in cell division. Furthermore, we will also explain the different stages of mitosis and meiosis. In the next few minutes, we will explore science for another moment. Do you know how cells reproduce? The cell is reproduced through cell division. Cell division is a process of producing a new daughter cell from the mother cell. There are two kinds of cell division, mitosis and meiosis. Today, we will discuss the comparison between mitosis and meiosis. Mitosis is a type of cell division that produces two identical cells with the same number of chromosomes. This type of cell division happens in our body cells. Meanwhile, meiosis is the type of cell division where a single cell divides twice to produce four cells containing half original amount of genetic information. These cells are the reproductive cells, sperm in males, eggs in females. In mitosis, there is one cell division happens in the parent cell, such as prophase, metaphase, anaphase, and telophase. While in meiosis, two cells division take place, the meiosis 1 and meiosis 2. In meiosis 1, we have the prophase, metaphase, anaphase, and telophase. While meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. In meiosis 1, prophase 1, the nuclear envelope begins to break down and the chromosomes condense. Centrioles start moving to opposite pole of the cell and a spindle forms. Importantly, homologous chromosome pair up, which is unique to prophase 1. Metaphase 1, spindle fiber attached to the paired homologous chromosome. The paired chromosome line up along the equator of the cell. This occurs only in metaphase 1. In metaphase of mitosis and meiosis 2, sister chromatids line up along the equator of the cell. Anaphase 1, spindle fibers shorten and chromosomes of each homologous pair start to separate from each other. One chromosome of each pair moves toward one pole of the cell, and the other chromosome moves toward the opposite pole. Telophase 1 and cytokinesis. The spindle breaks down and new nuclear membranes form. The cytoplasm of the cell divides and the two haploid daughter cells result. The daughter cells each have random assortment of chromosomes, one from each homologous pair. Both daughter cells go on to meiosis 2. In meiosis 2, prophase 2, the nuclear envelope breaks down and the spindle begins to form in its haploid daughter cell from meiosis 1. The centrioles also start to separate. Metaphase 2, spindle fibers line up the sister chromatids of its chromosome along the cell's equator. Anaphase 2, sister chromatids separate and move to opposite pole. Telophase 2 and cytokinesis. The spindle breaks down and new nuclear membranes form. The cytoplasm of each cells divide and 4 haploid cells result. Each cell has a unique combinations of chromosomes. The primary purpose of mitosis is to grow and replace worn-out cells, while meiosis reduces the number of chromosomes in the parent cell by half and produces 4 gamete cells. This process is required to produce egg and sperm cells for sexual reproductions. In this episode of Agham Alam Hub, you learn that mitosis is a type of cell division that produces 2 identical cells with the same number of chromosomes. In comparison, meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. Mitosis happens in body cells, while meiosis is in sex cells. That's all for today, Sci-Pals. Don't forget to like, share, subscribe, and turn on the notification bell. See you again next week for another moment. Only here in Agham Alam Hub, Palladian SciTech Portal. Bye!